organisms are more microbes than they are multicellular organisms. These microbes are also microbial communities are also very diverse. Just to give you an example, in some of the salamanders that I study, I found living on the skin thousands of different species, right? So it's a very diverse community. In addition, we know that microbes contribute significantly to the physiology of multicellular organisms. We know in humans there's been a lot of studies looking at connections with a lot of important human diseases. And in addition, microbes are the microbial community is very dynamic, so it changes throughout life, it changes across different species, and it changes across different environments. So these characteristics of the microbiome is something that makes us microbiologists super interested in understanding how they differ. And in particular, I'm very interested in understanding the connection between host, microbiome, pathogens, and how environmental factors influences these connections. And if you're not familiar with the amphibian literature, we know that several members of the amphibian microbiome are able to impede the growth of pathogens. In addition, hosts have a tremendous or some choice in what microbes can live on their skin. And we know the environment is also very important. So there's a lot of questions and a lot of very exciting experiments that we can do to understand how this relationship behaves. Particularly, one pathogen is very important for us interested in studying amphibian microbiomes is the amphibian chytrid fungus. There are currently two species of the amphibian chytrid fungus. There is BD, that is mostly infects frogs and is found throughout the world. It has almost a pan-global distribution. And then BISA, which has been currently or recently described, is right now its range is restricted to Europe and Asia, and it mostly infects salamanders. BD and B-cell or chytrid fungus is a very lethal pathogen to amphibians. And here we have two pictures, one of a healthy skin tissue, one that is infected with a fungus. And what the fungus does, it consumes the keratin on the skin, causing the amphibians to sloth, over sloth and lose this very important organ. And we know in amphibians, the skin is important for respiration and also regulation. So the fact that BD and B-cell disturb the skin makes them very lethal. But there are some good news, as I mentioned before. There are members of the skin microbiome in amphibians that are able to produce metabolites that impede BD and B-cell growth. And here is an example of it in action. Here we see some BD growing in culture, and you can see that there we haven't added any metabolites, or at least we didn't add any metabolites, and BD is growing happy, filling that space. But we can see that in the case where they added different metabolites from different bacteria, we can see that some bacteria are actually very good at impeding the growth of the fungus, where others might have some issues, but we do see that there's a reduction in growth. And this raises the potential to use some of these bacteria for probiotics. It's really exciting that we can think of ways to mitigate this disease. So I would argue that for us to be able to implement effective probiotic strategies and really understand how these beneficial microbes are helping that host, we have to understand the distribution and different factors that contribute to the distribution of beneficial bacteria across different hosts. And yet suppose that here we have a pool of all the beneficial bacteria that can live on a salamander. Well, we know, let's assume that these bacteria have the ability to distribute equally throughout environments. There are going to be several environmental filters that are specific to these environments, say humidity or temperature, that are going to dictate which bacteria can survive. And therefore, frogs or salamanders living in these different environments could be exposed to different beneficial bacteria in the environment. In addition, as I mentioned before, we know hosts have some say on what bacteria can colonize or not. There's a lot of work looking at phylogenetic correlations with community composition, so we can also assume that maybe aspects of the immune system could dictate what bacteria different individuals pick up from that environment. So in order for us to really understand how these beneficial bacteria are distributed, it's really important to evaluate the importance of these environmental and host-specific filters. And that's sort of what I want to allude to today in a study where we looked at terrestrial salamanders native to California. Again, we're looking at an aspect of their microbiome, and we were focusing on chytrid fungus. And as I mentioned before, B-cell is a recently discovered fungus. It thankfully has not been introduced into the United States, but we really want to stay ahead of that infection wave. If it does come in, we want to know, one, how are beneficial bacteria distributed across different species, specifically maybe species that we know are susceptible and others that are resistant. And then two, across the range of these species, what role is the environment playing in the distribution of these beneficial bacteria? So I want to do a shout out to an undergrad that collaborated with me in this project. This was part of our senior thesis. Shannon Witterberg, who is currently looking for grad school, so if you're interested, thumbs up on her. We sampled across two different families of salamanders, the longlist Plethodonids. We looked at the California slender salamander, which is broadly distributed and was present in a lot of our sites. We looked at the Ensatina salamander, which is another broadly distributed salamander in California. And then we looked at what we consider to be a susceptible family, which are the newts. And we know that at least one of the newts, Torica granulosa, has been tested for susceptibility, and we know that it's highly susceptible to the fungus. And then the other two, we're still trying to evaluate their susceptibility. We looked at multiple locations throughout the San Francisco Bay Area. We attempted to sample all species at each location, but the ranges really don't overlap. But we did make sure to include at least two species of the ones I mentioned before per location. So I'm going to briefly go over the methodology. I know we have a lot of 16S RNA sequencing research presented in this session. We collected skin swabs. We brought those into captivity, extracted DNA, amplified the 16S RNA marker. Then we sequenced those amplicons on a MiSeq. And we use Tryne2 to evaluate, characterize our microbial communities. A group in University of Massachusetts, Williams lab, has characterized a lot of bacteria. They have actually cultured a bunch of bacteria and tested their ability to impede BT in vitro. And they've sequenced the 16S RNA, so there's a very nice 16S RNA database out there of all these bacteria that are able to inhibit the pathogen. So what we did is we took representative sequences for our ASVs, blasted them against its anti-BD bacteria database, and that's how we were able to acquire a beneficial antifungal bacteria community. Then we applied meta-community structure analysis. This is a statistical analysis that would allow us to evaluate the distribution of bacteria across hosts and environments. And I'm not going to go deep into what meta-community structure analysis works. I'm happy to talk to you about that after this talk, but I'm just going to give you a brief overview. So whenever you do your 16S RNA sequencing, you have your OTU table or ASV table. You essentially have a table where you have individuals in rows and bacteria, right, species in columns. And what we do in meta-community structure analysis is we essentially have our table. We have ones and zeros in this case, based on presence or absence of bacteria. And then we ordinate our rows and our columns. We ordinate our rows, which are our hosts, based on community similarity, so that individuals that possess very similar communities are grouped together, and individuals that possess very different communities are grouped apart. And we ordinate our bacteria based on similar distribution, so that bacteria that have a similar distribution across hosts are together, and those that do not are set apart. And this creates this really cool pattern, I guess, so where we can evaluate an environmental gradient. And by environmental gradient, that could mean the environment on the host or the environment throughout the across populations. So this is we're able to evaluate how the distribution of these bacteria could be explained by environmental differences. And I'm going to give you some idealized. We compare this statistically to some idealized meta-community structure plots. And I'm just going to give you a brief overview of what is it that we could see and what's the ecological relevance, right? So here we have a checkerboard distribution, and what essentially is happening here is we have mutual exclusion. Whenever we have one bacterial species, there's another one's going to be absent. So that tells us about maybe the behavior or what's going on ecologically among bacterial species. Here's a nested distribution, so we can see that as we move up or down or up the rows, we can see that something's happening in the habitat where we have species loss, right? As we could be moving to maybe a habitat that's more harsh, that supports less communities, or it could be that some of these species are dominating the community. And here's a Clementsian distribution where we have a compartmentalization. What I mean with that is that as we move up and down these rows, we have a very strong signal of turnover, right? We almost have very distinct communities at these two extremes of that plot. Does that make sense? Awesome. Great. So I'm about to do what our advisors tell us not to do, is put a humongous plot. But I promise we're going to survive. I'm going to try to walk you through this. Oops, moving on its own now. So here, okay. So here on the rows, okay, what's going on? On the rows are all our salamanders, so I'm going to populate it with a lot more salamanders. And then on the columns are inhibitor bacteria. And what I've done, just to help you look at the abundances of these bacteria throughout populations, is I've colored the cells based on their relative abundance. If specific inhibitor bacteria is absent in the individual, we're going to have white color, and then if that bacteria dominates the community, there's a 1. So you can see that there are some bacteria that are pretty abundant here, and then others that are absent, right? So here's the entire plot, if it moves. Okay, so here's the entire plot. And what I've done here is I've colored the rows based on the genus that we sampled, on the host genus, okay? And there are basically 3 main patterns that I want you, or take-home messages that I want you to take. One is that if you look at the coloration, we seem to have a genus gradient, right? So down here we have Tarica, the newts. They tend to have a very different microbial or antimicrobial community than the Batrachosis, which on top of here, and then Ensatina sort of falls in between. So we have that really strong genus gradient. We do have compartmentalization, and if you notice that compartmentalization aligns with that gradient. So here up top, these Batrachosis have very different antifungal microbial communities than the Tarica on the bottom. And then in addition, the third point, okay, is that if we color the rows now by where we sample these salamanders, we do not have a very strong effect of location, right? You can see that now that gradient that we had before is gone because locality seems to not be explaining the distribution. So what does this tell us? Essentially what this is telling us is that these salamanders that might all be present in the same patch of forest, right, are all associating themselves with very different antimicrobial bacteria. And what this could mean is that if B-cell was introduced, we could expect that different bacterial species are going to be protecting them. And if there are any differences in their ability to impede B-cell growth, we could expect differences in susceptibility to the fungus. So just to go back to the questions, we know that different amphibian species tend to associate themselves with very different antifungal communities. In addition, environment has some effect, but not as strong as the species. And then I think this is super cool and super important because right now there is a big push to develop the probiotic therapy. So essentially what these results are telling us is that we might have to take a more individualized approach into how we treat these individuals. So essentially not one probiotic fits them all, but maybe think more about what bacteria could survive on a specific host. With that, I'd like to thank my funders. It would not be possible to do this without Shannon, who was great in the field. My advisor, Erica Brie Rosenblum, that has been a fantastic mentor, and then all the sampling locations and funding sources. And if I have questions, you guys can catch me up.